You've heard of high net worth, but we're talking about high net purpose. We want to find out how entrepreneurs and global leaders in our network are allocating financial, human, and intellectual capital in the pursuit of purpose. I'm your host, Joe McCarthy, and today we're in California with a truly captivating guest joining us. He's an inventor, entrepreneur, CEO, and the co-founder of multiple groundbreaking companies that in aggregate have achieved over 10 billion in public and private market capitalization. In our conversation, we cover the new age of medical discovery, including areas like generative biology, gene writing, and utilizing what Mother Nature has already given us in mobile genetic elements. This is the Geoffrey von Maltzahn podcast. Geoffrey, welcome to High Net Purpose.
Thank you. Thank you for having me.
We're sat here in Santa Monica, two of the tallest people in the area. You don't quite realize that you're the same 6'4" as your Texan self until you meet in person. But it's been good to listen to a few of your talks you've been involved in the last couple of days at the Montgomery Summit. And thanks for the time today to talk with us.
Of course. Likewise.
Okay, so straight in, have you got a articulation of your purpose?
I would take my purpose back about 20 years, and I think of it as to make biology engineerable. And that may sound confusing in that we've been using the word engineering in biology for 50 years or so, but we've never meant that we can build a cathedral and it will stand up on the first try. Or, the Sistine Chapel, or, variations of human creations in other realms. And we've sort of been guessing biology. Most of it works in ways that we don't really understand. But during our lives, that is all going to change. And the implications of that transition are almost unbounded. On the other side of kind of that guiding light, is a world of medicine where you never get sick. If you're sick, you have a cure for the disease or something that reverses the disease instead of just nudging it a little bit. It's a food system where fields across the world capture carbon dioxide and are net carbon sequestration technologies instead of emission technologies, and lots of other beautiful things.
And, we're talking about a revolution you think that's taking place in biological engineering today, and that's aided by computer power, and the main application area for you at the moment is medicine. Mm-hmm. And going back those 20 years ago, you were a chemical engineer to begin with, but then you moved to biological engineering. Is that correct?
Yeah, 20 years ago, I was an undergraduate at MIT. I went to MIT because I loved art and math, and I thought that it'd be a beautiful place to see how those two worlds might dance. I ended up sort of swapping art for biology, and sort of serendipitously, I was given a nudge to intern in a biology lab. 3 months later, I was mustering up the courage to quit, and my mentor sort of lit the guiding light that I described previously, that inevitably biology wouldn't just be, us squinting at gels and doing other laborious things to see how one puzzle piece might fit into the whole, but, we'd have a picture of how the majesty of biology really works.
And you got a PhD from MIT in biomedical engineering, and you won loads of awards. So were you that sort of wicked smart kid at school when you were younger? in Texas.
Yeah, I grew up in Texas. I've always been a really curious person. Love to learn, and I think in particular had this kind of lifelong affinity for kind of where the edge of knowledge is, and that can be a source of exhaustion for some. That's like a beautiful ramp up of enthusiasm for me. Like the worse our answers get in areas that are really important, the more excited I get.
The more you dig in.
Yeah.
And were your parents academics, or was it?
My father was. Father's an electrical engineer. he's also German. So I sort of think of that as kind of doubly creating a mindset that the whole world is a system of parts. And if you know how the parts work and how they connect together, you can predict the way the system works. My mother was a Montessori teacher, and there was a wonderful cognitive dissonance in our household in that, of course, she could see the world that way, but in the first instance, the world was full of dreams and hopes and stories and tales of adventure and shipwreck. And so in some ways, those two mindsets have kind of fueled my love for biology, in that at some point we'll understand, how to do the equivalent of land on the moon with respect to creating technologies in biology. We haven't been there. And, you have to actually love an adventure for the time being.
And because biology is a bit messy, right? It's not like when people think of engineering of machines and computers and stuff.
Well, it's messy because we don't understand it. So, we kind of like clumsy humans interacting with biology can make a mess of it really easily. But, in fact, biology is full of the most extraordinary technologies in the world. The things that we call proteins, for example, are the smallest and most amazing sensors, motors, engines, and devices in our entire technological realm. To stay with them for a moment, even cooler, they start life as a noodle and then fold themselves into those technologies. So like that'd be the equivalent of if this couch, this room, the television arrived as noodles and folded themselves into those technologies. That's happening trillions of times every second inside of you and I right now. So, I wouldn't describe biology as inherently messy, but, we don't have the dexterity with which to really elegantly interact with it right now.
It's a tough one to get our heads around, but we will get some practical applications in some of the companies that you founded. And because you've been responsible for co-founding companies that have an aggregate 10 billion in public and private market capitalization. And you sit as the CEO of 6 companies today. So trying to understand that, should we take a step back and look at you joining Flagship Ventures in 2009? And is that the sort of the umbrella for where your career has gone?
Yeah. So I started in undergraduate where that flame of where biology would go was lit. During my PhD, I got exposed to startups in a serendipitous way. And I just loved it. Like the rate of learning was incredible, the extreme partnership with other people. I was staying up all night for the startups I was working on, more so than other things that were in my life. And I started to imagine that if biology has the jump that I was describing a moment ago, then almost with certainty, the most impactful life science companies haven't yet been started, or they're just itty bitty today. And the reason for that is that if a technological advantage just sort of, slowly increases with a low compound interest, that probably facilitates the incumbents accruing, incremental gain. But when the rules change swiftly, which appears to be the reality we're living in, with near certainty, it's that subset of startups that have a perspective on the future that may seem speculative to the rest of the world, but to them is a fact. That when that fact turns out to be right and they make every decision on the basis of that, end up becoming, that next group of tallest trees. So I had just enough of a taste of startups to fall in love, and also to realize that they're these really hazardous, delicate things. And that, in a sense, if you want to be an entrepreneur, you're declaring 20 majors, or, you will need to have competence or mastery of a dozen plus distinct professions. And a lot of the advice that I received was, in hindsight, almost destructive. Like, the best way to learn is just go off on your own. And, that's okay advice in that many actually existing organizations can stifle people who are really entrepreneurial. But it's terrible from the perspective that if you were just trying to master one of those professions, your parents' basement and the internet wouldn't be the optimal educational platform with which to do it. So fortunately, I had a couple conversations with the team at Flagship, and they tempted me into a role where I'd be inventing technologies and starting companies internally. And the allure of that was, one, that was sort of my happiest space. But two, there was this combination of appreciation and kind of forever young mindset that the best was yet to come. And therefore, one shouldn't rigidly codify lessons of startups from biotech thus far, so as to be open-minded to, what the most extraordinary thing would need to be. And the second was that a hypothesis that Nubar had, which was that if you could bring 100 or so company founding experiences together in a team, you could eliminate a lot of the unforced errors associated with, startups dying of some diarrheal disease, that could have been preventable. But you could also create this, almost like sort of Olympics training for a championship, sort of mentality across a group where everybody's pushing themselves and pushing one another in concert.
So it sounds like an incubator model. Is that what it is? Or is it different?
We bristle a little bit at incubator, and that incubator is often snacks, foosball table, and other people's ideas in the mix. Instead, we're trying to sort of push ourselves into the unknown, like frontiers of science. Everything that we start now, we're the inventors of the founding technologies. And that has gone well. Sometimes people ask me why everybody doesn't do it. If it was also easy, then everybody should. This happens to be our sort of, special craving for pain or uncertainty.
And the provenance is Moderna is related to the group.
Yeah, Moderna is one of the companies that was founded out of Flagship. I wasn't involved in that, but Nubar and others were.
So some huge successes and a culture that allowed you the space to be able to do that type of much wider viewing of the world and what you could possibly achieve.
One of the most dangerous things in startups is that when you get going, you are silently committing thousands or tens of thousands of hours of your life to that. And if you appreciate that, then you should spend some fraction or multiple of that in advance of that moment. And we've got a combination of a beautiful culture and process across the team to try to put a ton of discipline in front of any idea so as not to grab onto something that is a good idea when, just around the corner is a great one, and just around the corner of that is something that could really change the world.
So do you generate the ideas, or do you talk to other? Okay, so it's not that you meet other MIT students who've got an idea and you work on it together, or are you the actual creator?
Most of the time it's us. And I would say we're anything, it's not like we're sort of turning off stimuli from the outside world. In fact, one of our fundamental beliefs is that the sort of apple falling on Newton's head kind of, epiphany moment is usually made up. Maybe Newton is an exception to that. But in fact, there's this almost lifelike evolution from a sort of insight that if thrown into the real world would become extinct quickly, but that if kept alive, through sessions of iteration between creative people who are, willing to follow a hunch and, voraciously and curiously and creatively, try to evolve that into something that could be extraordinary, really amazing things happen. So it's more of a team sport, and it's a more prolonged endeavor than people often describe.
Why don't we jump into an example of one of the great successes that you guys have had? Generate Biomedicines, so therapeutics, the intersection of machine learning, biological engineering, and medicine. where did that one start?
Sure. So we started Generate 6 years ago. Now that ChatGPT and other generative AI technologies are running amok in the world, it's a lot easier to explain. Somebody asked me recently whether Generate uses generative AI. And I said, yes, that's why we named it that 5 years ago.
So this is generative biology, which is similar in that you're looking at patterns and you're training the data in order for it to develop the outputs.
Yeah, exactly. So rather than go to town on all human language, mined on the internet and other sources, we've been trying to teach machines the languages of life. Those are, of course, DNA, RNA, and proteins as the engines that, make life possible.
Okay, we have taken a step back from there. Are you starting with the DNA sequencing and it's the data that's come from that you're trying to interpret more? Or
yeah, great question.
So
the flow of information in life is DNA makes, for example, an mRNA. That then serves as a short-lived template to one chemical amino acid at a time build a protein, which then, as I was saying before, does this beautiful gymnastic event of folding itself into a technology that either serves a purpose in life or is part of hundreds of billions of dollars of annual therapeutic sales that are proteins.
Okay, hold on one second. So we've got the DNA, so the mRNA is the messenger ribonucleic acid. I got there, the mRNA, which is then forming into the amino acid, which is a small molecule that's in the design of what the protein is afterwards. And that protein is what's then used in the therapeutics. Is that the step through?
So that information flow is how all proteins in our biology are formed. So whether it's somebody producing insulin, for therapeutic purposes, or whether it's you or I post a great workout in the gym, synthesizing new myosin within our muscle fibrils, that's sort of the universal flow. And what we started to imagine was that although the longer you're in biology, the easier it is to forget how profound it is that all of the properties of proteins, be they $1 billion therapeutic or be they an essential puzzle piece in our biology, are implicitly therefore present in the DNA sequence from which that protein came from.
Got it. Okay.
And if machines could figure it out, biotech would change forever.
So it's a computational problem, but it requires AI or machine learning or just a next advancement to be able to deal with that complexity.
Yeah.
And is that what the guys at Generate have managed to crack?
Yeah. So we started with two hypotheses that 6 years ago seemed weird. One was that maybe directing machines just towards every sequence of DNA that encodes a protein that we could get our hands on across every kingdom of life might give that machine a kind of safe Shakespearean mastery of how to speak or write the language of DNA. Second idea was that if one wanted to apply generative AI to therapeutics, it would be wise to try to figure out the most valuable thing first, which is all antibodies, all peptides, and almost all proteins provide one function in medicine, which is to bind to a target in a very specific location and not stick to other stuff. And if you could figure out what the DNA sequence of a molecule needs to look like to do that, then you might just have a radical impact on how every one of those therapeutics is created.
And traditionally, how would that have been tested? That's the trial and error methodology versus a supercomputer or a computer being able to do all those trials.
Yeah, so let's start with antibodies. Antibodies are most of biotech right now. And antibodies are these proteins that do an amazing job of sticking to a pathogen or binding to a target in a precise location. So you or I, if infected with a pathogen, our immune system would, make us weary while trying to figure out with trial and error the best solution to antibodies that would neutralize important proteins that are on the outside of that virus or bacteria.
Okay.
So just to take an antibody as one example, but it's true for almost every category of proteins, the number of amino acids that are involved in that antibody binding to its target is about 60. 20 amino acids that life can choose from at every single position. So the combinatorial number of potential antibodies is 20 to the 60 power, which is like atoms of the universe squared. So we don't get to use all the atoms of the universe when we find antibodies. In fact, it's way more marvelous that we ever find a solution than it is to imagine that none of those antibodies today are optimized. So whether it's our immune system discovering something or a lab, its search space is infinitesimally small relative to everything that's possible. And the power of using generative AI is that it can allow you to much more efficiently comprehend what the functional optimal would look like and make those predictions than anything we do right now.
And then, so the company's got a pipeline in oncology, immunotherapy, infectious diseases, and you work with big pharma, or you build your own, or what does that look like for that company, for Generate?
Yeah, a combination of both. So as I mentioned, 6 years ago, we thought this was a worthwhile pursuit, but who knows if it was going to succeed. Now we're convinced that 100% of protein therapeutics are going to be made this way, and there's going to be extraordinary advantages to doing so.
100% of therapeutics?
All of them. Yeah. And it's embedded in just, how little physics has allowed us to do, how small the search pace of trial and error-based approaches are, and, how as much as we enjoy the virtues of our human brain's capabilities, it's actually extremely limited when it comes to recognizing the patterns underlying how to program a protein, for example. So, simplistically, we have both an internal pipeline that we've prioritized, going after creating a set of medicines that we think have the potential to improve patients' lives by a lot. And we have a partnership with Amgen and a handful of academic relationships and others that are burgeoning.
And breakthroughs? Is there anything that you've seen so far in the work that they've done that you're like, wow, that is...
Yeah, I'll give you an example. This one's a couple of years old even. So imagine that this is sort of our like GPT-2 equivalent.
Right.
So we challenged the team a few years ago to take the top $50 billion of antibody therapeutic sales and generate antibodies that would hit same target, same epitope, which means the exact same spot on the target, same binding pose, comparable or better affinity, meaning it's the strength with which it binds to that, without going anywhere near the intellectual property estate of those parent molecules. In 3 months, the team did that for 100% of them. And by contrast, you wouldn't be able to do that in 10 years with a whole biopharmaceutical company's team and conventional approaches. I mean, that is like Robin Hood splitting the first arrow 50 times in a row with one that's, shot straight through it. And what that means is that something that has been incredibly difficult to do to date, we're able to find abundant sets of solutions for very quickly. And therefore, every single time some new target in biology is shown with an antibody or another therapeutic to provide a benefit to patients, there's the potential to find the best therapeutic for influencing that target. Many times, the first time something is demonstrated, there's some real liabilities to that as a medicine. And it's now clear to us that, we have the ability to quite substantially improve upon those first demonstrations and get great medicines to patients more quickly. It also opens up the ability to do lots of things that people haven't been able to do before.
Like what? Preventative medicine more, or?
Well, I'll give you an example with everybody's favorite acronym, COVID.
Yeah.
So the reason we get infected with COVID over and over and over again is the portion of the spike protein that does this Velcro-like step of binding to a protein called ACE2 has been changing a lot. And so your prior infection creates antibodies that then the new virus outsmarts. There are other portions of the spike protein that haven't changed almost at all, from COVID 1.0 to the wildest variants today. In fact, they haven't really changed from other families of SARS viruses. But our immune systems are awful at finding antibodies to hit that spot. Fortunately, generative AI is allowing us to. So we have antibodies that, as best we know, are the first to be able to hit those domains that haven't changed. And that might be a glimpse of one future category of applications for this, where we can take some of the most challenging pathogens in medicine and create therapeutics that they might not be able to resist.
Can we switch to Tessera?
Sure.
So this is into gene writing, which is different to gene editing and CRISPR. Could you maybe help us understand the gene writing component and how quick that's developed?
I'll start with the acronym CRISPR. So CRISPR is an amazing technology in Mother Nature's hands. It has been an extraordinary technology and has received the Nobel Prize within about a decade of its discovery. What it does is it's like a programmable set of scissors. It's able to grab onto a short RNA, use that to scroll through the halls of a genome, find a specific location, and then it cuts the genome in half. In bacteria, it's like a little immune system that chops up invasive DNA. When you do that in the human genome, our cells swoop in to try to fix it. They often make a mistake in the process, and that turns the gene off.
Okay.
So you should think of CRISPR as almost, the first tool that allowed scientists with ease to stroll through the halls of our genome and turn off light switches and figure out what they were controlling. So it has been revolutionary scientifically. There's a much narrower number of applications where turning things off permanently can provide a curative intervention for patients. Most of the time, if a patient is unfortunate enough to be born with a lifelong genetic disease, they're born with a mistake that has already turned that protein off. And therefore, to address that much larger category, you have to learn how to write DNA, either a small sequence or thousands of letters at a time.
So what would be examples where CRISPR works and where it won't work, and you need to get into the actual...
So if you have a gene that has gone haywire, turning that off is a good idea.
Okay.
But things break in biology similar to the ways that they break around your house. Most of the time, technologies in your house stop working, and unplugging them doesn't solve the problem. You need to fix or replace that function. And in DNA, that means making corrections or additions at the level of sequence. And CRISPR didn't evolve to do that. So the start of Tessera was the belief that in some ways, genetic medicine is going to be the final frontier of medicine at large. Like the best case scenario is that you never get sick, or if you have a genetic vulnerability to disease, that vulnerability is erased. DNA is going to be the rightful home for doing that. And so we went looking for machinery that might have spent billions of years learning how to write DNA in Mother Nature's lab. Those are called mobile genetic elements, and they're kind of like the smallest circle of life. So these are sequences of DNA that encode one or more proteins that can grab onto a copy of themselves and then write that into a new location. And in fact, they're so prolific that they built about half of your genome and about half of my genome over evolutionary time. So we started to imagine that if we could figure out how to program those, we might be able to turn that into a category of technology that could write DNA in a programmable fashion into cells around the body, thereby potentially opening up a whole new category of medicine.
So mobile genetic elements, how did you take that as the seed for the business? What did it, can you set the scene for when you all decided, right, let's go forward with this as the basis for the business, if that was the basis for the business?
Well, mobile genetic elements are the most abundant sequences in all of life. So not only did they build that much of the human genome, but chunks of the world around us of similar size. So you could conclude that Mother Nature gave more of her lab to mobile genetic elements than any technology in the history of life. which would mean that these would probably have benefited from more optimization and diversification than anything else. So we concluded that if writing in the human genome was going to be, in some ways, the technological feat that would open up the final chapter of medicine, then mobile genetic elements were the most robust foundation upon which to try to learn how to do that.
Got it. Okay. So it was a combination of the two elements.
Yeah. And we were struck with then which of these still work and didn't break down millions of years ago, which could work on the human genome. Of those, which could we program so that instead of writing their own code into our genome, which would mean there's something that can hop around, which is like a Jurassic Park style bad idea. Instead, they'd go to a desired location and write a cure into a given location.
So you were going to focus on doing the delivery, hit the focus area, and then make sure it was efficient and optimized. All of that, rather than doing just one element of it, was the goal of the company.
Yeah, we realized that if we were gonna build this company, we would need to innovate in at least two areas. One is learning how to gene write and borrowing and learning of machinery from Mother Nature with which to be able to make short modifications to the genome of cells to thousands of letters at a time. The second was, in order for these to work, they have to get into the cells that require a correction or the addition of function. And there have been some miracles done in medicine over the past 10 years with cells painstakingly being taken outside the body, having their genome engineered so that either immune cells can go after cancer cells or you can repopulate the bone marrow of somebody with sickle cell disease with cells that have been corrected. Both of those are really cumbersome. And the reason you've had to take cells outside of the body is we haven't had the tools with which to make those changes inside of the body. So we've put a lot of attention into inventing lipid nanoparticles akin to the kind of compositions that many of us have received in COVID vaccines, but they can go to cell types of interest in the body.
And that's the delivery mechanism.
That's right.
That's bringing it in. Okay. So you to develop that, and then you had to actually develop what was going to happen once it got into the cell. Mm-hmm. Was the bit that happens in the cell already kind of pre-defined, and that was the bit that was already solved, or that needed to be solved in a...
No, both were...
Because you got to do the editing once you get in as well.
Yeah, both were sort of leaps of faith. Wow. But we've been able to make some really amazing progress.
And when you say leaps of faith, were you starting from scratch, or you had something which said, no, we're going to be able to do this?
Well, we gained complete conviction that the existence proof of so much of our natural world having been built and written by mobile genetic elements meant that some variation of that technology was going to allow us to do things like cure sickle cell disease or other rare genetic disorders and write into immune cells and others. We had to figure out, what will the specific machinery look like that can perform that with the efficiency that's needed, with the fidelity that's needed, meaning making the change you want and not making a bunch of mistakes, and with which to get it to the cell types where those changes should be made.
So one thing we haven't touched on yet is that you've got a reputation for being able to build the teams. In this case, did you then have to go and find the right people to be able to take this forward? Or did you already have that within Flagship?
A combination of the two. In Generate, for example, Molly Gibson, Avak Kavejian, and a couple of other of our colleagues and I worked really closely in the early days. We then teamed up with amazing people, and now Generate's over 300 people. In the case of Tessera, Jake Rubens and I worked really closely at the outset. And one of the things I love about companies is if you establish a mission where you can see a better world in front of you and a technological path with which to achieve that isn't just, a lottery or guessing, but that you have real conviction around. It's amazing the kind of team orientation towards that mission that can come about. And, it sort of verticalizes everybody's learning curve. And, I fill your pockets with my hard-fought lessons and vice versa, challenge your ideas, and enjoy having mine be challenged. And it's one of the things that sort of led me into this world of startups. It's just the joy of seeing other people do amazing things and, trying to make space for that.
So lots of people talk about, mission-focused organizations, etc. But what does that actually look like? What are some of the day-to-day tools? I know that the overall purpose of what you're looking to achieve is huge in terms of human life. But how does that live on a day-to-day basis?
Yeah. So in the case of Generate, for example, if we're right, then an enormous fraction of future medicines are going to come from generative AI applied to protein design. And that's going to make medicines that are better, faster, less expensive, more accessible. In the case of Tessera, the word cure has been really carefully used for the past several decades, and for good reason. It's been almost impossible for current medicines to, deliver on that aspiration. That's about to change.
But don't medicines cure people?
Some do, but very, very few do. So like if you have a bacterial infection, antibiotics have been curative.
Yeah.
If you're hoping to prevent someone from ever having a heart attack, or provide an intervention post someone being diagnosed with Alzheimer's disease, or the vast majority of cancer therapies today, we have knobs which are able to nudge the disease in a favorable fashion, and sometimes we don't have anything. Much more frequently than we ever have something that can actually cure a disease. And in the case of genetic diseases, there's 300 million people around the world who are born with a mistake in their genome that gives them a lifelong sentence of disease. And if we're right about gene writing, the capacity to be able to provide with something as simple as a COVID vaccine, something that could allow a second grader with sickle cell disease to, with, single administration or a few of a therapeutic, live the rest of their life without sickle cell disease, is going to start to be possible. So that mission drives the way that we think about strategy on a daily, weekly, monthly basis. And probably more importantly, just, creates this amazing sense of purpose and commitment to try to, bring that into the world as quickly as we can.
But you've got a map to get there. You've also then got all the infrastructure around in terms of getting the financial capital to help support it and make sure you've got the best team to get there, etc. How in that culture is it still, people, you need high performance from people? It is very demanding, or is it different because you've got a lot more academics involved in the process?
Well, in general, we don't have a lot of academics involved in the process.
But there would have been the pretty highly educated people that worked in academics for a long time who are now stepping into a slightly different world in terms of being more commercial. And
number of PhDs, per capita inside the companies is really high. I think what the mission helps to fuel is, a sense of commitment and resolve in the face of constant challenges. Like this stuff, like everything in startups and biological startups, perhaps in special ways, is so hard. It's never easy. When it is easy, you have to make it hard, in that if you just take it easy when you're given this gift of something being feasible, and somebody else is going to bypass what you've done, and your contributions will have been negligible. So there's this, what I love about it is this sort of beautiful continual challenge to grapple with the uncertainty that surrounds a given endeavor, make the best decisions that you can on a continuous basis, make them swiftly, and joyfully recalibrate those when you realize that, you were operating with the wrong assumptions 1 week later, 1 month later. And, hopefully at the end of the many cycles of, scientific hypothesis to testing at different levels of the organization, in our strategy, in our science, will be the mission that we've been aiming for.
How do you prioritize your time and avoid getting distracted by, exciting new projects and ideas that may take you away from what you've already built and should be spending your time on?
Yeah, so you mentioned that I currently have responsibilities across 6 companies in a CEO role. The only reason that's possible is I have amazing people whom I think of my primary goal as being to help them have as much entrepreneurial impact as they possibly can, while ensuring that we're on a path to success. And so I think of my time allocation being in the zones that I'm most effective in, which is sort of in the scientific conceptual regime of what we're trying to achieve, in the technological details of where we really stand, and in trying to then iterate around dimensions of strategy to get to where we want to go.
Because I thought you were going to say the opposite. I thought you were going to say that you've been pushed away from the science a bit in the sort of role of CEO, and that there's other responsibilities within the companies.
There are, but at the same time, the only reason these companies exist is to do something that is scientifically special. So, you can't. That is like the essence of the company. All of the most important decisions are either like right in the middle of the science, or derived from what we're doing.
But you've got funding requirements, regulatory requirements, partnerships with pharma, all the rest of it coming through as well. But you managed to be able to get the time to still work in the science for that.
Well, in part because what I've tried to do is have aspired for me and members of my team to try to become the best in the world at starting with nothing and inventing a technology that might just change the world of biology. And in that regime, we often have no partnerships because were we to pursue partnerships, we'd activate eyebrow muscles on the other side of the table as opposed to enthusiasm because we haven't yet proven that beyond reasonable doubt, this is going to create new products and we're going to be able to do it repeatedly. And these are the kinds of diseases or aspects of our food system that have the potential to benefit from it. So that first phase of spectacular scientific ambiguity and uncertainty, that's like our happy place.
And is it a threat to big pharma, some of what you're doing in terms of their existing therapeutics?
Yeah, it's an interesting question in that in some ways one could say yes, but more likely it is going to be inevitable that these waves of change in medicine keep happening. Like today, the total market caps of companies that focus on variations of genetic medicine is closer to 0 than it is to $1 trillion of market caps. All protein therapeutics is about $1 trillion. All small molecule therapeutics is a few trillion. That's going to get flipped over the coming many years, in that the best case scenario is that you don't get sick. And the DNA driving every one of our cells around our body right now is the logical home for the therapeutics that most powerfully prevent that from happening. So from that vantage point, it's an amazing opportunity. Like the future rarely looks like the technological components of the past.
You asked some entrepreneurs today, what does greatness look like for you in the next few years? So posing the same question to you, what's the...
Oh, I love it. I am incredibly excited about the people with whom I work and some of the things that, some of the ways in which every year I feel like I'm meeting a new person because they're growing, so amazingly. And it's almost because of the achievements and growth and day-to-day challenges of the people that I have the chance to work with that we have the chance to do some amazing things. Some of those amazing things are, I believe we have a chance to deliver cures via Tessera to a large number of diseases that have either few or no treatments today. And that's a belief. At some point in the next few years, we're going to know whether that is reality. And one of the things that Moderna had said beautifully at their origins was that there would never be a Moderna which has just one drug. It's either going to be zero or a whole bunch. And so if we're right that gene writers can make these sorts of corrections, then we're going to be in the whole bunch category. And in Generate, we've brought the first 2 generative AI created antibodies into the clinic already. We're going to be bringing about a half dozen more over the course of the next couple of years. And hopefully we'll start to accrue the evidence that, in fact, there are much better drugs out there that we haven't been able to access before. And I'll give you one more thought, which I mentioned during the conference yesterday. One of my points of greatest inspiration in biology is that if you look at the number of potential proteins, for example, it's just one building block in biology. But if you freeze and focus on that unit, the number of potential proteins that could exist is 20 to the more than 100 power. I mentioned earlier, that's like atoms of the universe to some exponent. Even when you make really generous assumptions around what every protein that Mother Nature's ever been able to test in every life form that's ever existed, she's only been able to test less than 1 drop of water versus all of the Earth's oceans of what is possible. So, to think about it in simple terms, in that 1 drop of water is you, is me, all the people we've ever loved, every life form that we've ever interacted with, every life form that's ever existed on Earth is all in that 1 drop. And what I think is amazing about this transition of biology to being engineerable and the tools of AI that we're using at Tessera Generate and that are flowing into the field is that we're going to have access to those oceans. And the better mental model than that biology, has done all of these extraordinary things and we could tweak it a little bit, would be to imagine that all of biology is just at the very, very beginning of what's possible.
Wow. And that's what gives you optimism for the future.
Yeah.
I think you said before that you're genetically required to be, now that you're a father like myself. Last couple of questions. How do you stay healthy and well yourself with all of the obligations that you have?
Great question. Kiddos help a lot, both in terms of keeping me grounded and they're also like a Gold's Gym for some aspects of our psychology that we don't work out, during the day. In that, I feel like I need to abandon logic, deadlines, authority, just, put myself into the shoes of a 4-year-old and try to figure out what's going on. Physically, I'm lucky to have a couple people in my life who have really continually nudged me to believe that you can't be at your peak mentally unless you're vigorously challenging yourself physically. I have a trainer who's a former MMA fighter. He delivers like a year of pain to me within an hour on a regular basis. That pain is a beautiful way to set aside concerns of any given moment.
Okay, so some heavy exercise interspersed with some being a dad, where you lose all authority that you had in the office that day. What would you say to yourself, Geoffrey, going back to when he was just young? What advice would you give him?
First of all, the numerous imperfections of paths that I've taken have been on balance beautiful. So I wouldn't really want to be the interventionalist, voice from the future. But I think at every moment I've realized, that there really isn't a ceiling to what you should be seeking, in terms of the impact you can have on the world. Like, most of those are either created via our impatience or imperfections in our process and the way that we interact with other people. If you can patiently, vigorously interact with people that you love with a belief that the future can be better, it is astounding what can come out of that. So sort of took startups for me to realize how much more beautiful life is when it's a team sport. And maybe that insight sooner would have helped me.
What a great note to end on. Thank you for joining High Net Purpose. We will watch excitedly over the coming years as you build these great companies of the future. You're a busy man. Thank you so much for making the time for us today.
My pleasure. Thank you.
All content on High Net Purpose is provided as general information only. It does not constitute any advice, recommendation, or representations, and is not intended to influence listeners or users into making any specific investments or any other decisions. Please be aware that guests and presenters on High Net Purpose may have investments in any of the topics or products being discussed. Their reviews and opinions are their own and should not be taken as endorsements or financial advice. Before making financial decisions, we strongly recommend seeking advice from a qualified financial professional. This podcast may not be copied, reproduced, further distributed, or published in whole or part.